Hello everyone, welcome to Fun Molecule. So, in this series of R programming for biologists, I'm going to teach you how you can use R program for biological data management and analysis. So, in last video, we have learned some basics, functions of R and programs, how to write script, where do you get what, and few little examples. And in this video, I'm going to let you experience a little program of DNA sequence where we are going to translate our DNA sequence using RStudio. So with this, I want you to practice it with me so you will get a little confidence. And after that, in next videos, I am going to go with the plotting graphs and making data frames and how do we manage that. So let's get started with our program. So I want you to practice it with me. You will understand few of the things, and I will elaborate if you don't understand some functions. Okay, so let's get started. And I'm going to tell you when you use the hash key, and there will be no significant function or output for that. You can use it for your instruction data or a heading. You can use it like that to know your function, what you're going to write in your script, or what is the program about. So let's just say we are using this key and we are going to do the complementary DNA sequence analysis. And we can also write a sequence here, so when we are going to run we can just copy and paste. So you can do it with any sequence you want: ATCG, CGAT, GACT, GTA, and AD. Okay, that would be enough. And let's start with our program. DNA sequence. I would say whenever you are typing anything and you get a suggestion down the line, always click on the suggestion because it will lessen your error rate. So let's say we just did the DNA sequence. We are assigning function for it. Function, and that function could be X and the curly bracket. We use curly bracket when we are using block data, or we would say multiple statements and multiple set of data. When we are using it, we are going to combine it using the curly bracket. So the DNA sequence function, now base pair, will be equal to C and bracket, and that's A T comma C. Okay, so that would be our base pairs. And EPC, if we assign this any other letters, that would be unlist, and it would be structure split. That's structure split. Whenever you get a function, you can see why the structure split is used. It's an element of character vector. Split the element of character into X and substrings. Okay, so that's our structure split, and we are going to use 2 per for X, and that is for null. One more thing I want you to know, the null object, as you can see, you write anything and you will get the additional information about your function, the function you are giving. Also, anything, any mistake, any comma or bracket uh, that's not right could be an error, can create a bug, but never worry about the space. A space would never create an error. If you add space here or you don't add, that's not going to be a thing. So let's continue with our program. Paste the unlist and I'll apply. I want you to practice it with me so you can know where it could be an error, or if you face anything, then we can solve it. DNA sequence. Okay. Now we are adding one more curly bracket, because after that we are going to give the information what we want as our complementary DNA sequence. So the function would be if DNA sequence is A, the strings will assign it as T. If the DNA sequence is T, the strings will assign it as A. If DNA sequence is C, strings will assign it as G. If the DNA sequence is G, strings, we are going to assign it as C. Okay. If we are not done yet, if we have any other letters, if we have any other alphabets, we are going to give it as an exclamation. If DNA sequence is that's going to be percent and percent d square strings. uh, that will be assigned as n. If you have any other options, it will be shown as n in our sequence. So you'll know. Then return strings, and let's say now our program is done. So you want the results as collapse equal to the inverted comma, and that's it. That's our program. And what we are going to do is we are just going to select it from here to here, then click on run. And as you can see, the whole program will be in our console. Now we are going to give it, let's say, DNA sequence, and the sequence that we have already decided, it's going to be that. You can write any new sequence if you want. It's just paste it here. Now what we are going to do is, as you know, control plus enter, and as you can see, you get the complementary sequence of your given sequence. It's a small example. You can get the complementary sequence of whole gene or as much as you want, the big nucleotide sequences, and it will be as simple as that. And let's say one more example, DNA sequence. Here, as we have instructed, if there will be any other alphabets, the strings will translate it into N. So let's say A, T, C, G, G, C, D, D, T, T, D, V, G, J, then again A, C, G, E, G, C, A. Now, again, that's our input and control plus N. As you can see it here, you can see the complementary sequence and the complementary uh, N, where we have some different, alphabets. So I hope you have fun running this program, and I want you to practice it, and you can tell me if you have any doubt or if you get some bug. I will tell you how to uh, correct it, how to debug it, and in next video we are going to see how we can plot some graphs and out of some informations and the data frames, the tables, and how easy it could be. So stay tuned and practice. Thank you so much.